It's great.
Well, we need to make sure that at some point we cross paths on our travels.
We will in Seoul, latest.
Switzerland?
It's great. Not Switzerland.
Well, we need to make sure that at some point we cross paths on our travels. Well, we got to figure this out. Okay, it is 9:30 on a Monday morning. Welcome everyone to Neuroccino, where the rules is that everyone can present a paper, a book, or a piece of science they got excited about and tell us why. Today we have Marco. Marco is currently a rotation student with us in the lab and very excited about brain organoids, which is the topic of the paper today. And I also want to briefly mention that we have a CNS talk this week on Wednesday, 4 p.m. by Marco Palombo, talking about brain microstructure imaging. So do join us for that. Now, without further ado, Marco, if you want to share the link in the chat so everyone has access to the paper.
Yeah, sure.
And then share your screen.
Just a second. Here we are with the paper. I'm going to share my screen right now. Here we are. Just have to.
Don't worry, it's gonna be okay.
Yeah, just have to remove this. Okay, great. So, welcome everyone. Today I'm going to talk about brain organoids reservoir computing for artificial intelligence. So this is a paper that, evoked a lot of hype and a lot of articles in both bioengineering and neuroscience have been made on this project that just the paper has come out in January, in October 2023. Essentially, it is about merging brain organoids and neuromorphic computing. So, a little bit of background information. What are brain organoids? Brain organoids are 3D structures of neurons that are made from induced and pluripotent stem cells. In general, they use because they can mimic the developmental patterns of the brain, and also because among the vitro models, they can best represent what is a brain, and now it self-develop with self-patterns, and it can create connection. About neuromorphic computing, neuromorphic computing is computing method that wants to mimic the functioning of the brain in order to make it as naturalistic as possible. This is opposed to classic computing that is based on the fact that there are direct current patterns in bit. With neuromorphic computing, the activation patterns is a spike neuron. Okay. This is like a background information that will help you further to understand the paper because it's very difficult. So in this project, they wanted to use a brain organoid as a reservoir. Reservoir is considered as a processor of information. So you got a input pattern, and the input pattern is this computing, this neuromorphic computing system that decodes information. Then there is the brain reservoir, that is the brain organoid, that receives the input pattern from the multi-electrode array, and also the multi-electrode array, which is the framework in which the brain organoid is, will get the information and will decode the pattern that have been computed by the brain organoids. So why do they do that? Their essential reason was to establish a framework that can train a brain organoid to do some particular tasks, and also to establish the basis to build biocomputers. Why do they want to build biocomputers? Because they're energy and cost efficient. They're just think about the, like, here as an example, a human brain usually has an energy expenditure of 20 watts. When the current AI hardware that is based on artificial network, they need 8 million watts to have a comparable computational capacity fit. So what they did essentially, they tried to train brain organoid into do speech recognition. They had 8 participants with 8 speech characteristics that were regarding the pitch, the sequences, the special like the characteristics of the speech. And what they did was essentially they used the mil frequency spatiotemporal coefficient here to decode the input of the 8 different participants into a spatiotemporal sequence. And they inputted that into the brain organoid. So think that you have this multi-electrode array chip here and the brain organoid on top of it. And they used speech information from the participant to build this pattern of spatiotemporal activation, and they inputted into the brain organoids, and they assessed their brain activity. They did a multi-epoch training in which the brain organoid was trained on these different speech patterns that have different characteristics. And then they did the regression analysis. So essentially, this is a multi-electrode array chip here. Every square of the multi-electrode array has both the function of sending input, and this was based on the mil frequency substrate coefficient, and receiving input back and decoding information. So essentially, they made regression analysis that was based on this decoding, and they were according to all the characteristic of the response, which was spike count, spike timing, and spike patterns, and the weight of these different chips in the array. They were able to distinguish characteristic of the stimuli and characteristic of the response. And this is essentially a proof that the brain organoid could actually do computation and can be trained to do computation by giving input to him with the neuromorphic computing. And this is so exciting. And also they try to do the same thing. And this was essentially the same concept with, in this case, it was with predicting a nonlinear chaotic equation, the Eno map. So not to go too deep into that, but essentially, they wanted to predict nonlinear characteristics. So they did linear regression with these variables, and they still use 1D decomposition that is based on middle frequency step-aside coefficient in order to give naturalistic input to the brain organoid through the MAA. And then they train the brain organoid over different epochs in order to check for the capacity of the brain organoids to compute the Eno map. So, like, you don't have to know what is the Eno map, but it's just important to say that is a set of variables that predicts a chaotic nonlinear equations. And then they tried also, when doing this training, to insert a KMK2 blocker. KMK2 is used to is an essential chain that can induce long-term potentiation. So it is indeed a biomarker of synaptic plasticity. And when inducing this blockage of KMK2, the performance over this regression analysis in both speech recognition and linear regression of the Eno map was impaired. So actually, they concluded that this capacity to learn was dependent on synaptic plasticity. And yeah, so essentially, they proved that by inputting naturalistic input to a brain organoid and having a correct framework of decoding information, they were both able to use brain organoids as a computing framework and also to induce a patterned long-term potentiation. And here you can see, after the post-training, this is a degree of correlation between different networks in the brain, in the brain organoids. And you can see that here, the connectivity is very much enhanced by the training and the self-patterning. And that's it. So like, I can go deep into the system setup software and the framework, how to establish that, and also the middle frequency step-aside coefficient. That is a very, very interesting yet difficult topic to decode information into a naturalistic pattern to input the brain, but that's it. And I do believe this kind of research can be very important, both for biocomputers, which was the direct task that they want to implement, but also to check pattern of intervariability in synaptic plasticity between individuals. Because assuming that you can easily do brain organoids that are comparable between each other. There is no so much intervariability in developing them because this is a current problem that hopefully it will be solved. Then you can use lines from different patients. And after that, you can check for the capacity of the brain organoid to learn and the computational capacity of the brain organoids over time, which can be different between individuals. And this is obviously dependent on the feasibility and on the validity of the mill frequency substrate coefficient into giving a patterned inputs, and on the capacity early on of the brain organoids that usually they represent an early stage of development to show some computational differences. And that's it. That's it. This is my input. Now to hands-on. Happy to answer if you have any questions.
Thank you very much. Hands are already up. I think in order, Eva was first.
Hi, thank you for the presentation, Marco. And just a clarification question in case I missed it. So each Brainware is trained on one thing. They didn't try having one that was able to do several tasks.
They used this same brain organoids. There were 6 brain organoids in the supplementary data to do both the task. And then they assessed, but they were not cross-comparing different tasks in the organoids. They were just assessing the quality, and it was pretty, there was an equilibrium. I mean, there were not outlier in performance.
Okay.
Okay.
Thank you.
Mega has a question as well.
Yeah, thank you. Yeah, super interesting paper. Like, thanks for picking this. So I have 2 questions. For neural activity, are they just measuring like something like action potentials or like a sampled response? Like an
Yeah. So like this is very complicated, but in general, they assume that like artificial neural networks, when compared to brain organoids, it means that the brain organoids has to function as a network. Okay, so there is a pattern of firing between different hubs that in this case are neurons, and they generate output. And this pattern of output is actually the classification. Okay, it's part of the classification is the spatiotemporal resolution of these hubs that spikes out. About these characteristics of the stimuli that were assessed, they were spike count, spike timing, and spike patterns. These were the 3 that were used. But then you have to embed that kind of thing into a network.
Okay. Okay, and other question is how long these like organoids are viable? Like after they train it, how long they can use it before they die?
Yeah, so there is a whole topic on that because there have been studies trying to replicate, and I've just seen 2 months ago a conference in Italy when they were saying that they were able to retain brain organoids up to 8 months, which is crazy, but usually it doesn't happen like this. Usually after 90 days, they go into oxidative stress that is dependent on the fact that they outgrow it and don't have oxygen anymore in the center. And so they degenerate, and then this degeneration, it expands to all the remaining. So this is a limit, both to the size, but also to the capacity of the brain organoid to learn things. But good thing is that they can learn so much faster than the artificial neural network. And this was the time-consuming and energy consuming advantage that they have. After just 2 epochs, they can have a distinguishable pattern of recognition, which is not the case for artificial neural network, which needs to have, I would say, 100 of epochs in order to learn.
Okay.
Yeah.
So in their short lives, they're efficient, productive.
Yeah.
Okay. Thanks.
Eduardo has a question as well.
Yeah. So thank you again, Marco. Very dense and interesting paper. My question is about the kind of cells they use to build these experiments. If I got it correctly, the first task is a speech recognition task, right?
Yeah.
So did they use a specific organoids, and what kind of cells did they use? Because speech is fairly unique to humans, right? So
Yeah.
What's the trick?
Yeah, that's a good question. I also ask myself what kind of organoid they use. In general, cortical organoids are the one that you use for this sort of task. You cannot pattern the temporal part, of the cortical organoids. So they assumed that it was a right to just do a set of it was a generalistic brain organoid. There was not any kind of specialized pattern. So there were neural progenitor cells. They also had the glial cells, and then they saw this kind of self-patterning, cell circular patterning that they have. But there was no special characteristic except from the fact that there were cortical organoids and there were glial cells in them.
Okay, the cortical organoids from like human cells, right?
Yeah, human.
Okay.
Yeah.
Thank you.
Emma?
Just a sort of speculation question. With all this training and also what Eduardo is referring to of having specialized areas or cells, would you expect after training these organoids to see some kind of restructuring or?
Yeah. But this is what I was saying, and then I would love to show you. I would love to show you the supplementary material, but I have this bar that I cannot erase here. No, cannot. Just a second. Okay, I interrupt the sharing and then I will, okay?
While he's switching screens, can I just check, is anyone actually knowledgeable about organoids in this group? Has anyone ever worked with them? No. Great, we're all on the same page.
I've seen a couple of them once, but that's all.
This was what I was saying. So how they see the brain organoids, these are the brain organoids I was talking about. They are 5, not 6. I was wrong. Yeah. So like these are the pre-training, post-training, and the correlation delta, I mean, is a way to assess the strengthening on the of the connections. So there were actually structural changes, and you can infer that there were structural changes because inhibiting KMK2 led to the absence of training, absence of learning. And KMK2 is a main hub on the long-term potentiation that both create it enhances the number of receptors, postsynaptic receptors that are in the dendrites. and also it is sort of, it creates actin polymerization, which is one of the main markers of long-term potentiation, which is indeed strengthening and enlarging the synapse. So yeah, you can say there is a patterned long-term potentiation that creates structural and functional variation in the brain organoids.
Right. These graphs are functional connectivity, right?
This is functional connectivity, but they also talked about ChemK2. And ChemK2 refers to both functional and structural connectivity.
Right. Yeah, I was wondering more in the realm of if you did this in many organoids, if you would end up seeing some kind of pattern of optimal differentiation where, yeah, where all the organoids kind of tend to go towards, because that is sort of, I guess, how our brains also ended up.
Yeah,
evolving.
this is where they want to go, I guess. Yeah, and it's very cool. It's very cool. But like I guess that they have to be more precise, and the brain organoids have to be not reliant on their capacity to gain oxygen and not survive. And they have also to do a lot of stuff with neuromorphic computing, because it's not correct and precise at the moment. Not as much as artificial neural networks, but that's a good proposal. Yeah.
Thank you.
Chris?
The question.
Yeah, thank you for presenting that. I'm just a bit confused about the reservoir computing, because I mean, I just started getting interested in the reservoir computing, and as I understand it, you need a stable neural network, so the connections shouldn't change between the activity can change in the network, but the connections, the configuration of the network is not really supposed to change, as I understand it, in the reservoir. So I don't know if because here they show that the network itself is learning. But with the reservoir, I thought it was the layer after the reservoir that was supposed to learn from the activity of the reservoir. So I don't know if they talk about that, or if I just don't understand exactly how it works.
I don't understand either.
Okay.
Like it's a complicated thing, obviously.
Yeah.
So when I talk about structural changes, I can say that actin polymerization, it doesn't mean that there are new connections. Okay?
Okay.
Increase on receptor, it doesn't mean that there are new connections. But we are talking about brain organoids. Okay? So brain organoids go into self-patterning and exon self-patterning and spontaneous activity and this, you know? So it can be a big byproduct of the brain organoids to create these new patterns that is not dependent on the training, okay? Also, like you don't expect this kind of training to self-create patterns. You expect it to, you know, there are a lot of patterns already in the brain organoids. There is an overproduction of patterns and axons, and it is more likely than after the activity, some connection are cut off. You use or you lose, okay? So yeah, it was fixed in a sense, but
And do they
but I
compare
agree
it
with
to,
you.
I saw that they are comparing it to other models, like computer models, like LSTMs. But do they compare it to another, to a computer-based reservoir?
I will. Well, compared to a computer-based reservoir, they did that analysis, and they see that is still not there. But the computer reservoir has to do long short-term memory, has to do some characteristic that enhances its capability to process, and then it's better. And this was one limitation of the neuromorphic computing. But on the other hand, time, potentially, the fact that this brain reservoir, it is not affected by the von Neumann bottleneck. Don't know if you know it.
Nope.
No, it's just one mathematical concept that is based on the capacity to transfer information in a time frame. Okay, so like and it is an assumption of how computers are based in comparison to organic material. Organic material are not subjected to this von Neumann bottleneck that it limits the capacity to transfer information in a speed way, in a very fast way. Okay, and so yeah, like it's not yet there, but I believe that it will be. And it's a long talk to what are the conditions, but like I do believe that it will.
Yeah, no, it's definitely interesting.
Thank you. That's all the questions we have time for. So 2 announcements. We have a CNS talk this week on Wednesday, 4 p.m. Same place. And next week there's no Neuroccino because we're all on Easter break. So see you in 2 weeks for Neuroccino and on Wednesday for the CNS talk. Thanks for being here. Thanks for presenting, Marco. Bye. Have an exciting week in science.
Thank you. Bye.